Good afternoon, students. In the last class we discussed about spontaneous mutation, that is which is occurs spontaneously, that is naturally, whereas other type of mutation is induced mutation, okay, which is caused by the external factors, external environmental factors such as chemicals and radiation. Let us study chemical induced mutation. Okay. Although many mutations arise spontaneously, a number of environmental agents are capable of damaging DNA, including certain chemicals and radiations. Okay. Any environmental agent that significantly increase the rate of mutation above the spontaneous rate is called mutagen. Okay. They may ask in the exam, they may ask the question like what is mutagen, make note of it. Any environmental agent that significantly increase the rate of mutation above the spontaneous rate is called mutagen. The first discovery of a chemical mutagen was made by Charlotte Auerbach, who started her career in Berlin researching the development of mutants in Drosophila. During her research, she faced with increasing anti-Semitism in Nazi Germany. Auerbach emigrated to Britain. Okay, you might know all these things during Second World War, how Germany in Germany Nazi people were treated. Then she in Britain she continued her research on Drosophila and collaborated with pharmacologist John Robson on the mutagenic effect of mustard gas, which has been used as a chemical weapon in First World War. During Second World War, mustard gas was used to kill the human beings. Hence, the effect of these two scientists, they took study on what might be the mutagenic, what might be the effect of those mustard gas on human health. They studied this one. The experimental conditions were crude, since it was whatever the research they undertook, it was in secretly they were doing. Hence, the experimental conditions were very crude. They heated liquid mustard gas over a Bunsen burner on the roof of the pharmacology building, and the flies were exposed to the gas in a large chamber. Okay. After developing serious burns on her hands from the gas, Auerbach let others carry out the exposures and she analyzed the files. Okay. Then she analyzed the flies. Auerbach and Robson showed that mustard gas is indeed a powerful mutagen, reducing the viability of gametes. They showed that it reduces the viability of gametes and increasing the number of mutations has been noted in the offspring of the exposed flies. Okay. Because the research was part of the secret war effort, publication of their findings was delayed until 1947. Okay. Let us see what kind of the chemicals which cause the damage to the DNA. First one is the base analogues. All you know what is analog is. Analog is, it is a copy of one original molecule which almost looks like that original molecule, but it is not the original molecule. Okay, it is called as analog. Okay, one class of chemical mutagens consists of base analogues chemicals which with structures similar to that of any of the 4 standard bases of DNA. Whatever the DNA bases are there, A, T, G, C, the base analog, some of the chemicals base analogues were chemical, their molecular structures were almost similar to that of the 4 standard bases of the DNA. Then what happens if they are incorporated into the replication base analogs? If incorporated into the analogs, DNA polymerase cannot distinguish these analogs from the standard bases. So if base analogs are present during replication, they may be incorporated into newly synthesized DNA molecules. For example, fibromouracil is an analog of thymine. Fibromouracil is an analog of thymine. See, you can see here the structure of the normal base thymine, and the base analog of the thymine is fibromouracil. Here methyl group is replaced by the bromine. Okay, then it has the same structure as that of the thymine, except that it has a bromine atom on the 5 carbon atom instead of a methyl group. Okay, here it is methyl group instead of methyl group, here bromine has been added. Okay, then normally fibromouracil pairs with adenine just as thymine does. Okay, but it occasionally mispairs with guanine, leading to a transition such as thymine to adenine, leading to fibromouracil to adenine. Okay, which leads to fibromouracil guanosine, which leads to the cytosine guanine transition. Through mispairing of fibromouracil can also be incorporated into the newly synthesized DNA strand opposite guanine. In the next round of replication, fibromouracil pairs with the adenine, leading to another transition. Okay, see here normal pairing, that is fibromouracil, it usually pairs with the adenine molecule. Okay, mispairing of fibromouracil is which is anonized form will pair with the guanine. Okay, figure here fibromouracil, a base analog, resembles thymine, except that it has a bromine atom in place of a methyl group on the 5 carbon atom. Because of the similarity in their structures, fibromouracil may be incorporated into DNA in place of thymine. Like thymine, fibromouracil normally pairs with the adenine, but when ionized, it pairs with the guanine through Ubel. Okay. Okay, let us see the consequence of incorporation of fibromouracil in the DNA. Here, this is the normal DNA. When during replication, what happens? Strand separation takes place. In while replication, the fibromouracil in replication, fibromouracil may become incorporated into DNA in the place of thymine, producing an incorporated error. Here it is, incorporated error has been occurred. Okay, fibromouracil. Then what happens after during the next round of replication? What happens? This both DNA get separates, where in the next round of this guanine in the this guanine nucleotide pairs with the cytosine, leading to a permanent mutation. Here after next round of replication, this guanine pairs with the cytosine, which leading to the permanent mutation. Okay. Next, in here what happens when the incorporated errors of when the fibromouracil replication occurs, fibromouracil may mispair with guanine in the next round of replication. See here in the in the fibromouracil, the guanine is paired with the fibromouracil. In the next round of replication, what happens? If fibromouracil pairs with adenine, no replication error occurs. Here it is. Then this leads to the permanent error. Fibromouracil can lead to a replicated error. Conclusion is that incorporation of bromouracil followed by the mispairing leads to thymine adenosine to guanine cytosine transition mutation. Apart from the 5-bromouracil, another mutagenic chemical is 2-aminopurine, which is a base analog of adenine. Normally 2-aminopurine base pairs with the thymine, but it may mispair with cytosine, causing a translation mutation. Okay. Alternatively, 2-aminopurine may be incorporated through mispairing into newly synthesized DNA opposite cytosine and then later pair with the thymine, leading to a transition mutation. Thus, both 5-bromouracil and 2-aminopurine can produce transition mutations. In the laboratory, mutation caused by base analogs can be reversed by treatment with the same analog or by treatment with a different analog. Next, some other kind of chemical agents which brings about the induced chemical mutation are alkylating agents. Alkylating agents are chemicals that donate alkyl groups such as methyl CH3 and ethyl CH3CH2 groups to nucleotide bases. For example, ethyl methyl sulfonate, that is EMS, adds an ethyl group to guanine, producing O6 ethyl guanine, which pairs with the thymine. See here in the diagram, original base is guanine, and when it reacts with the mutagen ethyl methyl sulfonate, brings about the alkylation of the guanine molecule to which attaches the ethyl group to the 6 oxygen O6, which forms the O6 ethyl guanine, which pairs with the thymine, which pairs with the thymine molecule. Thus, EMS produces cytosine guanine to thymine adenine transitions. EMS is also capable of adding an ethyl group to thymine, producing 4-ethyl thymine, which then pairs with the guanine, leading to a thymine adenine to cytosine guanine transition. Because ethyl methyl sulfonate produces both purine pyrimidine and pyrimidine purine transitions, mutations produced by EMS can be reversed by additional treatment with the EMS. Mustard gas is an example of alkylating agent. Okay. Then next chemical agent which brings about the damage to DNA is the nitrous acid, which deamination, through deamination. Okay. Deamination can be induced by some chemicals. For instance, nitrous acid deaminates cytosine, leading to the creating uracil, which in the next round of replication pairs with adenine, producing cytosine guanine to thymine adenine transition mutation. See here, original base cytosine, which when reacts with the nitrous acid HNO2 brings about the deamination in the cytosine, leading creating the uracil. Uracil, when pairs with the uracil mispairs with the adenine, which brings about the type of mutation is cytosine guanine to thymine adenine. Then nitrous acid changes adenine to hypoxanthine, which pairs with the cytosine, leading to a thymine adenine to cytosine guanine transition. And nitrous acid also deaminates guanine, producing xanthine, which pairs with the cytosine just as guanine does. However, xanthine can also pair with thymine, leading to cytosine guanine to thymine adenine transition. Nitrous acid produces exclusively transition mutations, and because both cytosine guanine and thymine adenine to cytosine guanine transitions are produced, these mutations can be reversed with nitrous acid. Okay. Next chemical mutagen is the hydroxylamine. Hydroxylamine is a very specific base modifying mutagen that adds a hydroxyl group to cytosine, converting it into hydroxyl aminocytosine. Here in the picture you can see that original base cytosine, after reacting with the hydroxyl amine, brings about the hydroxylation and forms the hydroxyl aminocytosine, which pairs with the adenine and brings about the transition mutation of cytosine guanine to thymine adenosine. This conversion increases the frequency of a rare tautomer that pairs with the adenine instead of guanine and leads to cytosine guanine to thymine adenosine transition. Because hydroxylamine acts only on the cytosine, it will not generate thymine adenine to cytosine guanine transitions. Thus, hydroxylamine will not reverse the mutations that it produces. Next is the oxidative reactions, which brings about the damage to the DNA. Reactive forms of oxygen including superoxide radicals, hydrogen peroxide and hydroxyl radicals are produced in the course of normal aerobic metabolism, as well as by radiation, ozone, peroxides and certain drugs which metabolize in our body. These reactive forms of oxygen damage DNA and induce mutations by bringing about the chemical changes to DNA. For example, oxidation converts guanine into 8-oxy 7,8-dihydrodeoxyguanine, which frequently mispairs with adenine instead of cytosine, causing a guanine cytosine to thymine adenine transversion mutations. See here, when guanine undergoes when guanine reacts with the free radical oxidative free radicals, it forms 8-oxy 7,8-dihydroxy 7-hydrodeoxyguanine, may mispair with adenine. Okay. Then next chemical agents which brings about the damage to DNA is intercalating agents. Intercalating means the name itself suggests that it intercalates or it forms a sandwich like structure between DNA bases. Okay. Proflavin, acridine orange, ethidium bromide and dioxin are intercalating agent which produce mutations by sandwiching themselves, that is intercalating themselves between adjacent bases in DNA, destroying the three-dimensional structure of the helix and causing single nucleotide insertion and deletions in the replications. These insertions and deletions frequently produce frame shift mutations, and so the mutagenic effects of intercalating agents are often severe. Because intercalating agents generate both addition and deletions, they can reverse the effect of their own mutations. See here the structure of proflavin, and this is the structure of acridine orange, and how they are intercalate in the DNA. You can see in the diagram. See here, nitrogenous bases and intercalating agents. See here, these are the normal adjacent nitrogenous bases, and this one is the intercalating molecule. This is acridine orange, and here proflavin and acridine orange, they will intercalate. There will be space will be in between adjacent nitrogenous bases. They will go and intercalate, that is they get sandwich in between the nitrogenous bases, intercalate molecules. Okay. Intercalating agents such as proflavin and acridine orange insert themselves between adjacent bases in DNA, distorting the three-dimensional structure of the helix and causing the single nucleotide insertions and deletions in the replication. Apart from chemical agents, DNA also get damaged by the radiation. Okay, let us study how radiation affects the how radiation damage the DNA molecule. Okay, in 1927 Hermann Muller demonstrated that mutations in the fruit flies could be induced by X-rays. The results of subsequent studies showed that X-rays greatly increase mutation rates in all organisms. Because of their high energies, X-rays, gamma rays and cosmic rays are all capable of penetrating tissues and damaging DNA. These forms of radiation called ionizing radiation, ionizing radiation, which dislodge the electrons from the atoms that they encounter, changing stable molecules into free radicals and reactive ions, which free radicals then alter the structure of bases and break the phosphodiester bonds in the DNA. Then ionizing radiation also frequently results in double strand breaks in DNA. Attempts to repair these breaks can produce chromosome mutation. Ultraviolet light has very less energy, ultraviolet rays has less energy than that of the ionizing radiation and does not eject electrons and cause ionization, but it is highly mutagenic. Purine and pyrimidine bases readily absorb UV light, resulting in the formation of chemical bonds between adjacent pyrimidine molecules on the same strand of DNA and in the creation of pyrimidine dimers. Pyrimidine dimers consisting of two thymine bases called thymine dimers are most frequent. Apart from thymine dimers, cytosine dimers and thymine cytosine dimers are also can form. Dimers distort the configuration of DNA and often block replication. Most pyrimidine dimers are immediately repaired by some mechanisms, but some escape repair and inhibit replication and transcription. See here in the diagram, it is the normal thymine bases DNA molecule. When after absorbing radiation, after absorbing nitrogenous bases, the adjacent nitrogenous bases form covalent bonds forming thymine dimer. See how it looks in the diagram B. You can see here thymine dimers has been formed, distorting the normal structure of the DNA molecule. Okay, then when pyrimidine dimers blocks replication, cell division is inhibited and the cell usually dies. For this reason, UV light kills bacteria and is an effective sterilizing agent. For a mutation, hereditary error in the genetic instructions to occur, the replication block must be overcome. Bacteria can sometimes circumvent replication blocks produced by the pyrimidine dimers and other types of DNA damage by means of the SOS system. This system allows replication blocks to be overcome, but in the process makes numerous mistakes and greatly increases the rate of mutation. Indeed, the reason that replication can proceed in the presence of a block is that the enzymes in the SOS system do not strictly adhere to the base pairing rules. Hence, the result of this replication is that replication may continue and the cell survives, but only by sacrificing the normal accuracy of the DNA synthesis. Then SOS system is complex, including the products of the at least 25 genes. Okay, it is a gene. SOS system is a complex. It is a complex including the product of at least 25 genes. A protein called REC-A binds to the damaged DNA at the blocked replication fork and becomes activated. This activation promotes the binding of a protein called LEX-A, which is a repressor of the SOS system. The activated REC-A complex induces LEX-A to undergo self-cleavage, destroying its repressive activity. This inactivation enables the other SOS genes to SOS genes to be expressed, and the products of this gene allows replication of the damaged DNA to proceed. The SOS system allows bases to be inserted into a new DNA strand in the absence of the bases on the template strand, but these insertions result in the numerous errors in the base sequence. Then eukaryotic cells have a specialized DNA polymerase called polymerase eta that bypasses pyrimidine dimers. Polymerase eta preferentially inserts adenine opposite to a pyrimidine dimer. This strategy seems to be reasonable because about 2/3 of pyrimidine dimers are thymine dimers. However, the insertion of adenine opposite to CT dimer, cytosine thymine dimer, results in a cytosine guanine to adenosine thymine transversion. Polymerase eta is therefore said to be an error-prone polymerase. Students, so far discussed is the different methods by which DNA get damaged. So let us see how this how we can detect a chemical can cause a mutation or not. For this, to detect that carcinogenicity or mutagenicity of a chemical, one test is there, Ames test. It detects the mutation causing capacity of a particular chemical molecule. Okay, let us see how this experiment is performed. People in the industrial societies are surrounded by a multitude of artificially produced chemicals. More than 50,000 chemicals are in commercial and industrial use today, and from 500 to 1000 new chemicals are introduced each year. Some of these chemicals are potential carcinogens and may cause harm to the humans. You all know that carcinogen means it is a chemical molecule which has the capacity to cause cancer to the normal cell. Okay. One method for testing the cancer causing potential of chemicals is to administer them to laboratory animals such as rats or mice and compare the incidence of cancer in the treated animals with that of the control animals. These tests are unfortunately time consuming and expensive. Furthermore, the ability of a substance to cause cancer in rodents is not always indicative of its effect on the humans. After all, we are not rats. Okay, that means the whatever the results comes in the rats, whether if the chemical causes carcinogenicity or mutagenicity in the rats or mice, it doesn't mean that that chemical always causes the carcinogenicity and mutagenicity in human also. Okay, let us see. In 1974, Bruce Ames developed a simple test for evaluating the potential of chemicals to cause cancer. The Ames test is based on the principle that both cancer and mutations result from damage to DNA, and the result of experiments have demonstrated that 90% of known carcinogens are also mutagens. Ames proposed that mutagenesis in bacteria could serve as an indicator of carcinogenesis in humans. To conduct the test, Ames test uses 4 auxotrophic strains of the bacterium Salmonella typhimurium that have defect in the lipopolysaccharide coat. You all know that all bacteria contains pathogenic bacteria contains a lipopolysaccharide coat which normally protects the bacteria from chemicals in the environment. Furthermore, the DNA repair system in these strains has been inactivated, enhancing the susceptibility to the mutagens. See, the condition is that Salmonella type, whatever the strains you have considered here, they are they have defects in the lipopolysaccharide coat, which protects the bacteria from chemicals of the environment. Furthermore, the DNA repair system in these, if any DNA damage occurs in the bacteria, the DNA repair system in these strains has been inactivated, enhancing their susceptibility to the mutagens. Okay. One of the 4 auxotrophic strains used in the Ames test detects base pair substitutions. The other 3 detects different types of frame shift mutations. Okay. Then here for the Ames test they have considered only one strain, that is base pair substitution defective strain. Okay. Here each strain carries a histidine minus mutation. That histidine minus mutation means which the bacteria is unable to synthesize histidine, which render each strain carries a histidine minus mutation, which renders it unable to synthesize amino acid histidine, and the bacteria are plated onto the medium that lacks the histidine. Okay, this is the condition. Here they have considered histidine minus mutation Salmonella typhimurium, and they have plated, that means grown on a media that lacks the histidine. If the bacteria has to grow, they have to synthesize histidine. Okay, but the media don't contains the histidine. Since the histidine synthesizing gene has been mutated, they are unable to synthesize the histidine. Okay, only bacteria that have undergone a reverse mutation of the histidine gene, that is histidine minus to histidine plus, are able to synthesize histidine and grow on the medium. In the last class we have discussed about the reverse mutation. What is reverse mutation? From wild type to mutated strain, from mutated strain to again undergoing a mutation, forming a wild type is called as reverse mutation. Like that, the only bacteria that have undergone a reverse mutation, that can undergo a reverse mutation of the histidine gene, are able to synthesize histidine and grow on the medium. Okay, different dilutions of during this experiment what they did is, different dilutions of a chemical to be tested are added to the plates incubated with the bacteria, and the number of mutant bacterial colonies that appear on the each plate is compared with the number that appear on the control plates with no chemical. See here, in the experiment what happens, they have taken the histidine bacteria and what they have did is, during this experiment, bacterial histidine minus strains are mixed with the liver enzymes, which have the ability to convert compounds into potential mutagens. See what happens, some chemicals are basically not carcinogens. When they are enter into the human body, they then become, when they come in contact with the liver enzymes, they will become carcinogen. Okay, that's why they will add this bacterial strain. That's why they add these bacterial strains were mixed with the liver enzymes, which have the ability to convert compound into potential mutagens. Here what they did is, some of the bacterial strains are also mixed with the chemical to be tested for mutagenic activity. Here what they did is, bacteria are then mixed with the liver enzymes, and bacteria were plated on the medium that lacks histidine. They have added chemical, they have added bacteria, and they are plated on the media which lacks the histidine. Okay. Here if the chemical is mutagenic, it will brings about the reverse mutation in the bacteria and they will become histidine positive. See here, if the after incubation, if the colonies were appeared, after incubation, if the colonies were appeared, that indicates that the chemical mixed with that media brings about the reverse mutation in the bacteria. So the chemical added is a mutagenic or it is a carcinogenic. So when it is compared with the control plate which has not been added with any chemical, and the treated plates which is a which contains a carcinogenic chemical, it indicates that the chemical is whether a carcinogenic or mutagenic or the non-mutagenic. Okay, this is about the Ames test. Any chemical that significantly increases the number of colonies appearing on a treated plate is mutagenic and is probably also a carcinogenic. Some compounds are not active carcinogens but can be converted into cancer causing compounds in the body. To make the Ames test sensitive for such potential carcinogens, a compound to be tested is first incubated in the mammalian liver extract that contains metabolic enzymes. The Ames test has been applied to the thousands of chemicals and commercial products. An early demonstration of its usefulness was the discovery in 1975 that many hair dyes sold in the United States contained compounds that were mutagenic to bacteria. These compounds were then removed from the most hair dyes.